Ben Rich is also a master's student at University of Alaska Fairbanks, and he's going to be talking about his really interesting project on the Deshka and looking at pike diet. So Ben, if you are there. All right.
Hi, everybody. 1 second. Hi, everyone. I'm Ben Rich. I'm a master's student here at the University of Alaska Fairbanks. And today I'm going to be talking about my research with Northern Pike on the Deshka River. First, I'd just like to say that I really enjoy the symposium, and I feel like the great strength of it is that it's a bunch of people coming together who really care about these places and these fish. And for that reason, I'm going to try to go through my presentation kind of quickly and leave plenty of room for comments because I really want to hear from you and get feedback on what other people think this project can do for these fish in this place, and to involve you in this discussion. So first, we have a couple overarching questions. We're focused on Deshka pike specifically, but we think there's broader implications to this work. We're really interested in how invasive species in freshwater systems respond to climate change. And the Deshka is a great system to look at this. Pike are an invasive predator, as Taylor pointed out, and they've been in the Deshka for a long time. We also have really good high detail water temperature data, as Becky said in her presentation earlier. So it's a unique system and an opportunity for us to look at these relationships. We're also very interested if warming water temperatures will shift predator-prey relationships and how that dynamic might play out with pike and salmon in the system. And then if suppression is really worth the time and resources. Suppression is kind of something that we feel the need to do in these contexts of invasive species. And sometimes it's worthwhile to take a step back and look at our impacts and if we're really doing what we think we're going to do. So more applied questions for the Deshka specifically. Are predation rates of pike on juvenile salmon changing? Is the risk of pike predation equal for juvenile salmon across the watershed? And are there specific areas in time when pike are really targeting juvenile salmon? And are those the times that we should be trying to suppress them? So a little background on the Deshka. The Dene name for it is the Tuquentu, and it was a seasonal home to the Deshka hata. I hope I'm pronouncing that correctly, and somebody please correct me if I'm not. There was a large village site at the mouth of the Deshka. It's been culturally important for a long time. It has a long history of human habitation. It had large historic runs of Chinook that were very important to people living in this area going back time immemorial, as well as people in this audience today. Also, the system is non-glacial and slow moving, and it's tannic and warm. And I'll direct your attention to the pictures there on the right. As opposed to some other systems that we like to look at, the Deshka is really dynamic. It's not all slow moving warm slough habitat that we think of often when we think of pike. It has areas that are steeper gradient with faster moving water that are not conducive to pike, as well as areas where the floodplain stretches out and there's good habitat for pike. So objectives of this project, we want to analyze how pike diet has changed in the last decade, and it varies over space. And we also want to look at water temperature profiles in these sloughs. As Becky pointed out earlier, we have a really good idea what water temperatures are doing in the tributaries and the main stem of the Deshka. But some of these sloughs are kind of different. They are old river channels that have been cut off, and some of them have upwelling. And just from some of the point measurements that we took this summer, we learned that some of our assumptions about them might not be correct. Some of them seem to be colder than the adjacent main stem. So we're really looking forward to getting some of our temperature logger data back and characterizing those sloughs in their temperature regimes so we can compare that. We also want to model predation under different climate and water temperature scenarios, and we'll do that through a bioenergetics framework. And field proof and improve an intrinsic potential model that I'll talk about here in a second. Our specific goals for this year, we wanted to get on the ground in the Deshka, catch as many pike as possible, and take diet from as many as we could. We also wanted to improve our netting and capture techniques and work out a method that would stand up for future research to comment on whether or not pike are present or absent in a given location at a given time. So our sites that we selected, the map on the left is our sites overlaid on that intrinsic potential model that I was mentioning earlier. So a previous grad student, Chase Jalbert, took LiDAR and some aerial mapping and looked at watershed characteristics. And he looked at the elevation, the gradient, or how steep the stream is, as well as floodplain width. And by combining these three factors, he was able to identify areas that have wider floodplains and are better for pike habitat, and areas that don't really have that off-channel habitat. So for our sampling, we focused on the areas of high intrinsic potential, where there is a lot of off-channel habitat for those pike. In the map on the right, you can see that we spread our sample sites out over a long distance. The Deshka is about 32.5 miles long from its confluence to where it dumps into the Susitna, and we spread out our sites across the entire length of it. Previous sampling in the Deshka has focused on the lower 5 miles, which has been suppressed, but the upper river has never been suppressed and sees very little sport angling pressure. So I'm really excited to see our results from there. So what we did, we ended up capturing 521 pike total this summer, and we sampled in the end of May, June, July, and August. So we had 4 sampling events. And our sampling was pretty well spaced across space and time. If you look at our upper figure, that shows our main sites. We had 6 primary sites, as well as some kind of incidental ones that we sampled when we had the opportunity. And our catch was pretty consistent across sampling events. It decreased a little bit in our last one, but we tried to replicate our effort as best as we could and keep that consistent between events. Also, our catch was pretty well spread out across the river. As you already saw from how our sample sites are dispersed, you can see a gap between about river mile 7 and 15. That's that long area that doesn't have much off-channel habitat that you could see in the intrinsic potential model. So diet, still need to get into kind of the nitty-gritty of the diet data and analysis, but their diet was really diverse. It largely consisted of soft-rayed fishes, so that's pretty much any fish other than stickleback and sculpin. So the stuff that goes down easy. And from the literature, we know that's what pike will usually target if it's available. Salmon was also apparently important seasonally. We saw that salmon were more present at certain times in certain locations. Going to do further analysis to see if there's any kind of predictive quality to that in models. And we also saw lots of lamprey across sampling units. So to give you a better visual of what these pike are eating, we took all the stomach contents from a sample of pike. Those bins on your left, those are probably from 20 to 30 fish. So you can see it's not a whole lot of contents, but they're eating a large diversity. Some of the larger fish are eating full-size rainbow trout and other large prey items. You also have a bunch of juvenile salmon, invertebrates, lamprey, and even the occasional mammal. So the size structure of the pike, despite having a larger sample of over 500 fish, what we saw was really consistent with Taylor's sample of 30 fish from the Deshka, mostly smaller fish. And also comment to Bruce's question earlier, we really didn't see that many large fish in the system, and also cannibalism was exceptionally rare. We saw less than 5 cases of cannibalism in all of our diets. Another component that we worked on this summer was presence and absence. So we wanted to go to a site and be able to comment with a quantifiable level of confidence, whether pike are there or not. And we tried a new sampling tactic for that. We tried pairing eDNA with these new fancy backpacks and electrofishing. And we still need to go and analyze those samples, but I'm really excited for those results. And then we paired visual counts and gill netting. And still have to analyze the results from both of those, but we're hoping that going forward, either of those methods and or other methods from the literature we haven't tried will allow us to go to a site and be able to tell with fairly good confidence if pike are present or not. So our next steps, detailed analysis and modeling. We do full bioenergetics modeling with temperature data from our sloughs. And we also need to work through that eDNA and visual count data to assess those methods. And I'd really like to acknowledge our field crew. They work tirelessly in the field for us. I was also fortunate enough to have half my committee in the field with me this year, capturing pike, which is awesome. Our volunteers and Fish and Wildlife staff, super helpful with eDNA support, as well as funding and supporting this research. And with that, I'd like to open it up to questions and comments. Love to hear what all of you have to say.
Great, thank you, Ben. Looks like we're getting a couple questions, and feel free to put those in the chat. All right, so Kevin Fraley, I'm sorry if I'm pronouncing that wrong. Hey Ben, nice work. Did you find any particular gill netting technique most effective? Example, blocking off a slough mouth. Did you catch many other fish species in your nets? You showed the pike eating some mammals. What kinds were they eating? Oh, I'm excited to hear that again. You had some
record-breaking stuff. Hey Kevin, thanks for the question. Okay, so for gill netting methods, what we ended up doing was we're moving through the system in short time spans with canoes, so we need to be really mobile. So instead of putting out gill nets and letting them soak for a couple hours, we're using active netting methods. Earlier in the season, before all the weeds grew in, we were basically seining with gill nets. We would start near where the slough connected to the main river and work our way towards the back, and that worked pretty well. Later, when the weeds filled in the sloughs, we used a different method. We would visually observe where pike were congregated in the slough using a canoe or walking the shore beforehand. And then if there's a pocket of weeds where pike are really sitting in, you'd surround it with nets, and then we'd go in there with canoe paddles and scare them out into the net. Both those were pretty effective. And I think largely because we were so active and monitoring our nets, we had very little gill bycatch. We caught a cisco or two, I think some round whitefish, a couple suckers, but our bycatch was very minimal, and they're all really easy to release in good condition. As far as their eating mammals, we saw both mice and shrews represented. Didn't have large numbers either, and they were often pretty digested.
Great. Let's see, we had a question from Bruce. Lamprey were important in your pike, but the lamprey population is way down a little now. I'm interested in what differences lamprey have on salmon and or pike success.
Yeah, I'm really curious about that too, Bruce. It's interesting to know that the populations are down.
Yeah, that is interesting. Andrew said, did fish tend to move out of the sloughs and into the main river during times of low river flows?
Oh, that's a good question. Kind of hard to comment on. Yeah, when the water was lower later in the season, some of our smaller sloughs seem to have fewer fish in them. As to where they went, that's anybody's guess. Does anyone else have any thoughts or questions?
Sorry, I was talking to my mute button. That's good. It looks like there aren't any more questions.
Okay.
But it sounds like both Ben and Taylor are open to anyone reaching out, so feel free to do so later.